Neurons are beautiful, and also a bit mysterious. For a long time, scientists were unable to visualize the brain like we can today. Instead, scientists observed the macro structure of the brain and had to make assumptions about what was going on inside. In 1871, a German scientist named Joseph von Berlach observed under the microscope that the brain is composed of one continuous structure, an interconnected and rather fluid, mushy interior, where regions are connected through a web of intracellular signals. This theory, the reticular theory, really caught on when it was endorsed by the great scientist Camillo Golgi, who helped gather evidence on the continuous neuron theory. This sort of made sense given the technology he was working with, and it would explain why and how the brain can contain thoughts and share them throughout its continuous internal structure. Turns out, they could not have been more wrong. With somewhere around 300 billion discrete neurons, the brain is a complicated mess of signaling. Neurons talk to other neurons via a physical gap, about 20 to 40 nanometers wide, and let me tell you, we are nowhere close to truly understanding how it all works. Although recent technology has rapidly aided in our ability to see what's going on in the brain, new technology is coming out all the time, and there's still so much to learn by simply visualizing neurons. There are synapses to be explored, circuits to be mapped, and trillions of glue-like glial cells waiting for discovery. There is so much left to see in the brain. So, how can we do that? And oh yeah, what's DiI, the topic of this video? DiI, short for that, is a lipophilic cationic indocarboxyanion dye that is usually made as a perchlorate salt. It is applied to a cell membrane and allowed to diffuse laterally until the dye covers the entire membrane. It can be used in either fixed or live tissues, and it has a variety of applications, but it is particularly useful to visualizing neuronal axons. What should result is a singular illuminated neuron, which can be visualized using fluorescent microscopy. So with that, I'm ready to take a shot at doing it on my own. DiI stains can be bought as a solution, paste, or solid crystal, and it comes in a variety of fluorescent colors, such as red-orange, far red, or even green. The DiI available to me is actually DiO solution, which means that it will fluoresce green, but I'm going to continue referring to it as DiI throughout the video since that's the standard. Here's the mouse brain I'll be using for today's procedure. It is perfused with 4% PFA and left in the fridge overnight. It's important not to leave the brain in PFA for longer than 24 hours because that could result in damage to the cell membrane, making it unfit for DiI staining. I read that a single strand of hair is a good way to get a really precise application, so I started out with that, but it ended up being a pain in the ass. So I went with something that I had on hand, which was a plastic micropipette. Turns out this might have been too much and not as precise as I needed because I ended up being able to see the DiI spot with my naked eye, so I'm not sure how accurate this will be. While waiting for the DiI to diffuse, I put it in a warm water bath in order to encourage cell membrane fluidity. Typically, we store brains in the fridge to prevent mold and degradation, so setting these slices in the warm water bath is taking quite a gamble, especially for leaving them as long as I had planned to leave them in. I really had no idea how long to leave the slices in the incubator, but some sources were hinting at as long as a month. I ended up leaving it in for 3 weeks. After the 3 weeks were up, things were not looking so good. The slices look rather mushy, and I'm worried what I thought would happen, mold, has happened. But I went ahead and started prepping a slide anyway. Mounting was going to be tricky. The slice was supposedly 400 microns thick, and a cover slip would not be able to stick to the slide. I started fashioning this parafilm-lined slide, leaving a gap in the middle for the brain to sit. Hopefully, this added height would help prop up the cover slip against the thick slice. I started getting ready to remove the brain and place it on the slide, but literally as soon as I touched it to the water, the whole thing started to dissolve. The brain was just gone. I panicked and tried to fix it, but there was honestly nothing I could do at this point other than make more of a mess. The more I touched it and tried to fix it, the worse it got. So, at this point, I decided there was only one thing left to do: throw it in the trash and start all over again. This time, I was going to be a lot more careful about letting the brain mold. So instead of using a fixed tissue, I used a live tissue. This should allow the DiI to diffuse faster than mold can grow. I also decided to use a glass micropipette to apply the DiI in hopes that it would be more accurate than a flimsy plastic pipette. All in all, this round should turn out a lot nicer and take just a fraction of the time. I wanted to get a minimum time required in the books, so I let the slices sit for only about 6 hours and then mounted them for visualization under the microscope. Looking around, I didn't see anything too exciting until I scrolled across this strange looking web of tissue. It doesn't look like cell bodies or anything I normally study, so this was enough to intrigue me into taking a closer look using a confocal microscope. Looking at that same region using a Nikon acquisition software attached to a confocal microscope, I was actually pleased with how this looks. The dye didn't diffuse along any axons, but seeing as I only let it sit for 6 hours, this is pretty good, and you can see it diffuse across some of the dendrites. I imagine letting it sit for a little longer could make some really pretty pictures, so I was excited to try out this procedure again. I didn't want to change so much for this round, so I followed pretty much the same protocol. The only difference here is that these tissues are a little fresher. I also let it sit for closer to 12 hours. This increased amount in both time and dye to really help me reach my goal of staining a full axon. This one turned out a lot brighter than the first one. I didn't enhance or brighten this photo very much, and I was using a relatively low power laser. Honestly though, I think it was too much. The microscope is picking up everything, and all these little non-neurons are just noise to me. You can see a little bit of directionality, indicating that it did stain some neurons, but again, they just didn't go very far. I know it wasn't because I had too little dye, since I had really overdone it on the dye, so it seems to me like I have to let the brain sit for pretty much as long as I can before it starts to grow mold. This time, I'm going to use a dissecting microscope to help me apply the dye. I also took off my crazy nails, and some more DiI arrived in the mail. The yellow was a few years old, so I'm excited to work with something a little newer and hopefully a little better. Also, this one will fluoresce in more of a red spectrum, so I'm excited to see that color under the microscope. Working with the dissecting microscope has already been a huge help, and I honestly don't know why I didn't think of it before. Now I can actually see what I'm doing. Oh, and side note, this is not how you hold a pipette, but it gave me a lot of control, so I think the position of this dye will actually be the best. However, again, I think this might have been too much. When it's easily visible to the naked eye, I tend to get a bit worried about what I'm going to see under the microscope. But I'll leave it rocking in the fridge for the night and see how this one fares in the morning. The mounting is definitely getting a lot easier with practice, or maybe it's just my lack of acrylic nails. Now, this is strange. It's bright, like way too bright, which I think might be due to the fact that I used the same protocol of a 4-year-old stain on a brand new stain. But it's also strange because these aren't neurons. It's more like clumps, and while I'm glad something happened, I'm going to need to take a closer look to see what's actually going on. So, it's certainly labeling something, but what it is really hard to tell. It's also incredibly fluorescent. I have the laser power on a fraction of what it's normally at. This region is known as the thalamus, a relatively dense and fibrous internal region of the brain, so I think these strange tendrils might be an artifact of the thick tissue that holds neurons in place, which is not what I want to see. I want to visualize small dendritic projections, so seeing as the red dye has the potential to become its own project with its own set of challenges, I think I'm going to go back to the DiO for the next round because I was closer to success with that one. I want to note that this process of staining live cells doesn't start with a fresh brain slice like I show. It starts with a live mouse. Now, I won't show the decapitation or any gory stuff, but in a future video, I'm planning on showing the behind-the-scenes process of how I take the brain from its body and into these incredibly small slices. So if that's something you're interested in, please consider subscribing to my channel. I love to bring people into the world of the lab and have so many projects that I just can't wait to share with you all. But getting back to the slices at hand, I'm in the process of preparing the brains on a slide so they can be visualized on the microscope. They've already sat in the fridge overnight, and you'll notice that I'm not just doing 1 or 2 slices this time. This is because I feel pretty confident in the protocol and don't feel like any of these will be a waste or just practice. With 4 slices, I'm pretty confident that at least one of them should have the staining in the right spot with the right amount of dye. But I guess that's really up to the microscope to show me. Ah, hubris, the villain to progress. These slices did not turn out as expected. Something happened, and I don't know what, but the majority of them didn't take up any of the dye. Like the fluorescence is there, I can see the green, but it's not in any distinguishable shapes, meaning it didn't diffuse into the membranes, except for this good old slice second from the left. When I scrolled across this, my hope for this round was completely restored. I can see that there's so much going on, and I literally ran to the confocal to take a better look. So there's a lot of noise, but these are definitely neurons. The larger clumps are cell bodies, and the thinner lines are the dendrites coming off of them. I'm having a hard time finding full and clearly labeled neurons though, so I'm going to briefly switch to the eyepiece view since rendering on the software takes a long time. This screen recording is actually sped up, so switching to the eyepiece means I can quickly scan the whole slice without having to wait for each image to render. There's a lot of cool structures, but I came across this one neuron which just radiates in all directions. And I really want to get a closer look. It already looks super cool on the eyepiece, and I wish I could look at it longer, but consistent fluorescent light on a sample actually makes the dye fade, so I'm going to try to be quick in order to preserve the color. Yep, that's the neuron. I know it's sort of hard to see with all of the noise, but I want to direct your attention to the bright circle in the lower middle region of the screen. That is the cell body, and as I scroll through the vertical axis of this cell, you can see the different angles that the many dendrites take as they radiate from the center. It reminds me of a spider, except with way more than 8 legs, and they just come off from all different directions. In a 3D view, you can sort of see how complex the neurons actually are. I feel like a lot of the time neurons are portrayed as being very orderly and possessing just a few meaningful connections. But that's not true. Connections can change, and connections are messy. As neurons learn, they position themselves like contortionists in order to get the most function out of its many extending limbs. And it's honestly so beautiful. I'm really excited how this region turned out. Another structure I want to pause at are the hollow-looking cells in the upper left corner. Recall that DiI stains the cell membrane, not the insides. So that means when we get a two-dimensional view of a cross-section near the middle of a cell body, we are looking at a region where the DiI couldn't reach, the inside of the cell. So it looks hollow because this is where many organelles live, in this 15-micron wide oval in the center of a neuron. So my process was by no means exact, and I'm still trying to tweak a few things like the actual technique of putting on the dye, but for now I think I'm going to wrap up the YouTube side of this project and call it good. I'm really happy with these pictures, and any future iterations on the project would just be small tweaks to the final product. However, if I do end up mastering the procedure at some point in the future, I might do a follow-up video to really demonstrate the incredible uses of DiI, because this is only the beginning.